Hi everyone. In this video, we are going to pursue our journey inside the skeletal system, and we are going to talk about bone development or the process of ossification, meaning how soft tissues in the body can be converted into bones. We have two types of ossification or bone development in the body, endochondral ossification and membranal ossification or membranous ossification. We will start by the process of endochondral ossification. Endochondral ossification means that we have the body makes a model of the bone, the outline of the bone in the form of fibrous tissue. And then that fibrous tissue is converted into cartilage, and then that cartilage is converted into bone. So you start by soft tissue, like fibrous tissue, that is let down by fibroblasts, and then the chondroblasts or the immature cartilage cells will convert it into a cartilaginous model. And then on top of that cartilaginous model, you will have deposition of calcium and phosphorus. Now, this is one example of cartilaginous tissue as seen under the microscope. And the cartilaginous tissue, just like the bony tissue, is formed of a protein matrix, that homogeneous purple substance that you see in between the cells. And then you have the immature or mature cartilage cells, the chondroblasts or the chondrocytes that lie inside spaces called the lacunae. And then the chondroblasts or the chondroblast starts to lay down proteins that give the matrix this rubbery consistency that gives you the cartilage. So actually, cartilage is one type of connective tissue, but a specific type of connective tissue that is tough and flexible. During intrauterine life, when you are still in the uterus of your mother, and during the second month of intrauterine life, much of your skeleton is made up of a cartilaginous model. And then during the third month, so until the beginning of the third month, during the first 2 months, your skeleton is actually made of cartilaginous pieces of cartilaginous organs that are later on be or will be formed into bones and will undergo ossification. During the third month, the cartilaginous tissue will be invaded by osteocytes coming from the blood vessels that come from the periosteum, and the osteocytes will the osteoblasts at first will start depositing minerals, and those minerals will lodge or be deposited in the matrix around the chondroblast, and therefore the matrix of the cartilaginous tissue gradually gets ossified and gradually becomes converted into bony tissue. And this is what we said before, that the blood vessels coming from the periosteum include three main types of cells: the fibroblasts, to lay down proteins like collagen and elastin that are called fibrous molecules because they are elongated, thin molecules, and they make a fibrous model of the skeleton. And then we have the chondroblasts that come again from stem cells in the periosteal blood vessels and that lay down this rubbery matrix that appears blue under the microscope because it takes hematoxylin stain. And finally, we have the osteoblasts. So all the three cells come from stem cells in the blood. But the fibroblasts work they work first and they form the model. And then the chondroblasts work next and convert the fibrous model into a cartilaginous model. And finally, we have the osteoblasts that convert the cartilage into bone just by depositing calcium and phosphorus and mineralizing the matrix. So in endochondral ossification, we start by a nutrient artery. And this nutrient artery comes from, as a branch of the artery, the longitudinal artery that runs along the periosteum. how does the nutrient artery reaches this fibrous model, or this group of cells that will form the bone, via a foramen or an opening that will be later on called the nutrient foramen, and we have seen it in the shaft of the bone before, and the shaft of the bone is called the diaphysis. So there is actually a place through which the nutrient artery reaches the tissue model that will be converted into bone, and that opening will later on persist in the shaft of the bone or in the diaphysis of the bone, and will form the nutrient foramen, because it's a foramen or an aperture or an opening that helps entry of arteries and therefore of nutrients. This nutrient artery and its branches will invade a certain part of that model of tissue that will form the bone. They will invade the central part of that model that is rich in stem cells, and that is called the primary center of ossification. And this artery is bringing in osteogenic cells. By osteogenic cells, we mean generally the cells that help in the formation of bones. And those cells include the osteoblasts, the immature bone cells that will lay down minerals. But before the osteoblasts, we have the chondroblasts. They are not mentioned here, but we also have the osteoclasts. And we said before in the previous videos that the osteoclasts are bone-eating cells that can erode any unnecessary parts of the model, whether they are bony parts or cartilaginous parts. So actually the osteoclasts at different stages of bone formation are important for remodeling. Just like we have a primary or essential center of ossification in the center of the diaphysis, we also have another center of ossification in each of the epiphyses, or the rounded heads of the bones, and they are called secondary ossification centers. So endochondral ossification is actually a process that occurs slowly, in which there is gradual replacement of cartilage by bone. The cartilaginous matrix is being mineralized, and calcium and phosphorus are being deposited. So how does this occur? The slow conversion of cartilage into bone by the deposition of minerals onto the cartilaginous matrix, and that's why we call it endochondral ossification. Ossification means formation of bone. Chondral means cartilage, and endo means inside. So inside the cartilaginous matrix, we will be depositing calcium and phosphorus. So most fetal cartilage that was present during the first 2 months of pregnancy will be eventually replaced by bony tissue. But some cartilage remains in the body without ossification. And this persistent cartilage stays for a certain purpose, because the cartilaginous matrix, as we said before, is rubbery and flexible because it contains specific proteins that give it this consistency, and therefore cartilage will remain in the cartilaginous surfaces between the bones, which we call the articular cartilage, because they are related to the joints between the bones. So articular is the adjective, which means related to the joints. So as we will see on the next slides, the ends of long bones are covered by cartilage, and this cartilage is called articular cartilage, and it gives smoothness to the ends of the bones to prevent friction, especially in joints where there is constant movement, like the elbow or the knee joint, and especially in the joints that are weight-bearing, like the hip joint and the knee joint. But we also have some cartilage at the end of the nose, at the tip of the nose. So the tip of the nose is cartilaginous in the outer ear and in the cartilaginous rings in the trachea. So the outer ear is cartilaginous and the end of the nose is cartilaginous, or the tip of the nose is cartilaginous. Here you can see a cartilaginous joint, for example, the knee joint. So you can see that the epiphysis of that upper bone and the epiphysis, I think this should be, or maybe the femur, but the femur has like condyles here. Suppose it's any bone and this is another bone, and the site where the end of one bone is in direct contact with the end of another bone is called the joint. Here you can find cartilage. So this cartilage was essentially cartilaginous, and it did not become ossified. And this is called the articular cartilage. And you can see here the connective tissue that surrounds the joint. And that is called here, this is the connective tissue that surrounds and protects the joint. It is called the joint capsule. And lining the joint capsule from inside, you have a membrane that secretes fluid, and that membrane is called the synovial membrane, and it secretes fluid, which is called the synovial fluid. The presence of fluid in between the articular surfaces is a second factor, a second important factor to prevent friction in the joints. So friction between the ends of the bones inside a joint cavity is prevented by the smoothness of the articular cartilage and by the synovial fluid that is rich in hyaluronic acid and in other substances that make it viscous and that lubricates the joint. Here we can see the steps of endochondral ossification in the form of a diagram. So here you have the hyaline cartilage model. The primary center of ossification starts to appear, and it's rich in stem cells, which are undifferentiated cells that can divide very rapidly and that can differentiate into any other type of cell. And then the blood vessels penetrate this primary ossification center and bring in more stem cells that will be converted into that will differentiate actually into fibroblasts, chondroblasts, and then into osteoblasts. So bone starts to deposit around those blood vessels and to form trabeculae, as we said before. This is the primary center of ossification that receives an artery and a vein, of course, for giving nutrients and oxygen and carrying out waste. At the same time, we have the secondary center of ossification, where ossification also occurs in the heads of the bones. As you can see, there are certain parts that are not ossified. So we have the articular cartilage that is not ossified to keep it smooth for for preventing friction in the joint. But we also have another very important part of the bone, which that transverse part of the bone, which is not ossified, and that is called the epiphyseal plate. The epiphyseal plate is like a linear section of the cartilage that stays as it is, and that is not ossified at the beginning, and that separates the epiphysis from the diaphysis. Why is this epiphyseal plate important? Because if this epiphyseal plate becomes completely ossified like the rest of the bone, elongation of the bone or growth of the bone in length would stop. So the epiphyseal plate is considered the growth plate for the bone. As long as it is not ossified, the cartilage cells divide and give more thickness to the epiphyseal plate to elongate the bone, and then gradually parts of that plate become ossified, but always leaving a part of cartilage for active growth and increase in length. When the epiphyseal plate becomes completely ossified, we say that the epiphyseal plate is now closed and that the bone is no more capable of elongation. So in endochondral ossification, we have a cartilaginous model, as we said, that is gradually replaced by bone. And by the end of the 8th week, about 2 months, the chondroblast or the immature cartilage cells start laying down a cartilaginous matrix to form a hyaline cartilage model. The hyaline cartilage is very characteristic under the microscope, and it looks much like this diagram that I showed you. It's refractal like glass, and that's why it is called hyaline cartilage under the microscope. And then the chondroblasts, after having led the cartilaginous rubbery matrix, they get trapped inside spaces, inside pockets that are called the lacunae. I forgot to tell you that meanwhile, this cartilaginous model is surrounded by what we call the perichondrium. It's fibrous tissue. So tough fibrous tissue. We always start by fibrous tissue, and then the fibrous tissue gets cartilaginous, and then the cartilaginous tissue becomes ossified. But in the perichondrium, it stays fibrous. It does not undergo any further development. So every cartilaginous model, even after the whole fibrous model has been converted into cartilage, the perichondrium stays fibrous as it is with all its blood vessels. And this is what will be converted later on into periosteum around the bone. And the periosteum, it also stays fibrous and does not become ossified. As you know, all these cells originate from stem cells. When the chondroblasts find out that they are trapped inside the lacunae, they start resorbing the cartilaginous matrix. They start swallowing the cartilaginous matrix. They hypertrophy, they increase in size. Of course, when they increase in size, they need more energy. And since there isn't enough energy that reaches them, they will die at the end of the day, because even when the nutrients that reach them are high, yet they cannot cope with the hypertrophy or the huge increase in size that occurred. The death of the chondroblast is good news for the bone, because this means that there will be enough space for the osteoblasts to invade the lacunae and start depositing calcium. When calcium deposits around the chondroblast, this leads to further death of the chondroblast, because it like chokes them. It prevents nutrients from reaching them. Now we still have the perichondrium that is still fibrous, that is not converted into cartilage nor into bones, and it still contains stem cells. So some of the stem cells in the perichondrium become differentiated into osteoblasts. Those osteoblasts will be very important because they start laying down calcium and forming compact bone around the cartilaginous shaft. And as we said before, the place where all this process of ossification starts is the primary center of ossification. That includes blood capillaries, stem cells, and of course, these stem cells will be converted into osteoblasts. They will invade the central part, which is the primary ossification, or that will be converted into the primary, or considered the primary ossification center. This group of capillaries containing young immature osteoblasts that invade the core of the cartilaginous shaft to prepare for the formation of the primary ossification center is called the periosteal bud. And then after having established this primary ossification center, the osteoblast will migrate upward and downward to extend the process of ossification to the rest of the bones. And by the end of the 12th week, about 3 months of pregnancy, we will start the same process of ossification, but this time in the heads of the bones or in the ends of the long bones, which are called the epiphyses. Now the bone needs a medullary cavity or a bone cavity, so the bones refer to the osteoclasts to eat up the central part of the bony matrix and form a cavity, a long cylindrical, hollow, continuous cavity in long bones that will contain the bone marrow, and that will be surrounded by, as we said before, a layer of a spongy bone that has its own cavities full of bone marrow tissues. As we said before, the whole cartilaginous model will be calcified except for the articular cartilage and for the epiphyseal plates. As I've just explained, this is like a wrap-up of what we said. The epiphyseal plate still contains chondroblasts, and the chondroblasts will divide to cause elongation of the bone. If the epiphyseal plate becomes completely ossified, then bone elongation will stop. But there is another type of ossification that is called the intramembranous ossification, meaning that in flat bones in particular, like in the ribs, in the scapula, in some flat bones of the skull, as you can see, and some bones of the face, what happens actually is that there is no cartilage formation. There is a tissue model that looks like the final bone, and deposition of calcium and minerals occurs directly into this membrane. So we move on from the tissue state, the fibrous tissue state, directly to the ossified state without having the cartilaginous stage in between. And you can see that the vault of the skull, or the upper curved part of the skull, contains here, you can see 4 bones of the skull, and they fuse together through joints called sutures. And in the skull, the suture lines can be seen where the different plates of the skull appear, and they are joined together. The process of intramembranous ossification will be explained in detail in the second video.